What's up, baby bio penguins? So, number one, I have a whole bunch of hiccups. So if I just hiccup during the thing, just kind of let it slide. But we are going to do a Unit 2 review. My students have their exam over Unit 2 tomorrow. So they asked me if I would do a live stream. So thus here I am doing the live stream for my students over Unit 2. So I have a little PowerPoint that I put together just with like the visuals for us. And I'll put this all onto my Weebly, so you'll be able to access this on YouTube and all that later on when you're studying for the AP exam, or if you are a student who isn't currently at Unit 2 yet. So hi to my actual penguins and hi to any other students that are out there. So real quick, what is Unit 2? So Unit 2 is kind of, we're looking at surface area volume ratios, we're looking at our organelles, we're talking about like membrane transport, and then for my students I put cell communication in here, and then there's 2 FRQs that I figured I would discuss with y'all, because just, I think we need to look at some FRQs and how some FRQs apply to this content. Like, so a lot of stuff, so a lot of this stuff may, be, knowledge based, but you're going to have to be able to apply a lot of it. So there's some stuff that I won't be able to tell you exactly how you're going to see it on the AP exam. Do I know when I'm going to have my 2021 stuff ready? I am building my 2021 stuff currently. So it's a rotating wall of me making things. So let's get diving into this. So first thing, surface area volume ratio. Okay, so this is the formula sheet. Okay, so you're going to notice that you have to be able to solve either the sphere or a rectangular solid or a cylinder or a cube. So my students, any of these are fair game for our unit exam that we have tomorrow. Anyone else in the world, any of these are fair game for your AP exam in May. So I have seen on previous exams there has been a sphere before, and there has been a cube before. I have not seen a cylinder yet, and I haven't seen a rectangular solid, but that doesn't mean that they don't exist. It doesn't mean that it won't show up this year. So that's what I've seen so far. So let's just do a quick example. So my students have already done a cube in class, so I figure my example could be a sphere. So if we have this sphere, and we look, of course, our surface area is 4 pi r squared, and our volume is 4/3 pi r cubed. And if we go ahead and do this math, okay, assuming that r is 3, so my radius is 3. So then if I go in and I say, okay, well, let's sub that in. So surface area is 4 pi r squared, so sub in that 3. We know that 3 squared is going to give you 9, so 4 pi r, and so leave it as pi, and so we have 4 times 9, which should give you 36, assuming I know how to do math at this moment. And then if we do our volume, of course, we have 4/3 pi r cubed. We sub in our 3, so that would be, I skipped a step. So 3 cubed should be 27, and then whenever you multiply 27 by 4 and then divide by 3, you get 36. So our surface area to volume ratio for this should be 36 pi over 36 pi, which just gives you 1. And so the big thing about surface area volume ratio is that they're going to try to ask you about which cell is most efficient. Hello, Panda. They're going to ask you which cell is the most efficient. And so your cell is going to be most efficient when it has the highest surface area to volume ratio. So if you were to solve a different sphere and that sphere gave you, surface area volume ratio of 3 versus this one that had a 1, the other cell would have been more efficient because the more surface area you have. I always tell my students like doors, right? So the more doors that you have to get in and out of your cell or your city or whatever you want to say, the more capability there is for materials to move across that membrane, and you can get more nutrients in and more waste out. Also, the bigger the cell, the more space it has to travel in order to get across. Think about whenever you spray some air freshener in a room, it takes forever for it to fuse across that whole room. And so thinking about the cell, you've got to get these materials that are dissolved in your cytosol to move across your cell. It's going to take a while. So the smaller your cell, meaning the more surface area to volume ratio, the better for that cell and the most efficient it's going to be, which is why we are multicellular, because of the fact that our cells are broken up into multiple different cells, so that we can be efficient and big. And of course, if y'all have questions, go ahead and you can put them into the chat as we go. But again, I'm focusing on Unit 2. So if you have questions that are not Unit 2, I will probably be ignoring you because this is designed to be for us to focus on Unit 2 like exam that my students have tomorrow. So somebody says, what's a cell? A cell is the smallest unit of life. So within the cell, you basically all life started from this one cell. We all know that our lives started with a zygote. So sperm and egg came together, making the beautiful zygote that then went through mitosis that then formed you. And then there are some cells that are going to be single cell, like bacteria is a single-celled organism. Amoebas are single-celled organisms. A student just asked if they're going to have the formula sheet tomorrow. You do not get the formula sheet, but any formulas that you do need, you can ask me for, and I'll give them to you. So I'll give you the surface area formula, and I'll give you water potential if you need that one. Where was I going this? Your first block, which means that you're going to be the first ones that determine whether I didn't give it to you or not. So if you're missing something, be sure to let me know in first block. Otherwise, first block just won't get the equation. Third block will when they remind me about it. Okay, so let's look at our organelles. Okay, so before we go into this, remember that with the organelles, it's going to be more application based. Okay, so they'll give you a specific example of some disorder that might be occurring, and you have to figure out which organelle was had an error, or if they told you that there was an error in this organelle, what would be the repercussions of that error. Okay, so looking at this chart, this is the exact same chart that my students have seen multiple times now. So the nucleus is where we're going to have all of our hereditary materials. So that will be where our DNA is and our chromosomes are. They condense down, of course, using histones, which are proteins. Okay, this is also the site where we're going to have RNA synthesis. So within our nucleus, you're going to have this enzyme called RNA polymerase that will read your DNA and will then make an RNA, and then that RNA will leave the nucleus to be able to go through another process called translation. So our ribosomes are going to be the site of this translation. So this is where we're going to have our protein synthesis. There's two different types of ribosomes you can have. There's free ribosomes. Free ribosomes mean that they're just free floating in the cytosol. These cytosolic ribosomes are going to make cytosolic proteins. So your free ribosomes that just chill out in the cytosol make proteins that just hang out in your cytosol, and they're part of your cytosol. So like relay proteins in cell communication, proteins that are used for different processes within your cell, all that is made by those free ribosomes. Okay. You could also have bound ribosomes. Bound ribosomes are the ribosomes you're going to find on your rough ER. These are the ones that will make membrane proteins and proteins for secretion. And so it kind of depends on where they are and what kind of protein they're going to make. All proteins start out as free ribosomes until they get a signal that then causes them to bind, make the protein, and then they'll detach again. And we'll get through more information on how ribosomes work when we get into transcription and translation. At this point, just understand that ribosomes are going to make proteins, and then understand that cytosolic ribosomes make cytosolic proteins, and that membrane-bound ribosomes make membrane-bound proteins and protein for secretion. Ribosomes are considered an organelle. They're not a membrane-bound organelle, which means that your prokaryotes and your eukaryotes both have ribosomes. So when you're kind of thinking about the different types of cells, ribosomes are going to be found in all cells. Okay, so going back to our list. Our rough ER, as we said before, is going to be protein synthesis for our membrane as well as secretion. So that's because of the fact that it is studded with those ribosomes. Smooth ER is going to be used for lipid synthesis and detoxification. You also find that it is going to have the calcium storage. So calcium is a secondary messenger, and it's actually stored within the smooth ER. So when the cell gets the signal to release that calcium for the secondary messenger, that's when it gets released out of the smooth ER. So mitochondria, and don't you dare tell me it's the powerhouse of the cell in this AP exam. I'm watching you. So the mitochondria is the site of ATP synthesis. Energy cannot be created or destroyed. So it's very important that you don't say that like we're creating energy in the mitochondria. All we're doing is we're using the energy from glucose and we're breaking it down, releasing that energy and storing it in the form of ATP. So we are going through ATP synthesis, and it's the cytosol respiration. More specifically, it's the site of the Krebs cycle and the electron transport chain. Glycolysis is going to occur within the cytosol, so you don't really need to worry too much about that at this point in Unit 2. But as we get deeper into the curriculum, you'll kind of learn the different places where things happen. Somebody asked a question. Yes. Somebody says, are membrane-bound organelles only in eukaryotic cells? That is a true statement. Prokaryotes will not have any membrane-bound organelles, which is why they have no nucleus, versus our eukaryotes, so your animal cells and your plant cells are all going to have membrane-bound organelles. So basically, all the organelles we talked about are all going to be in a eukaryote, and I'd say very few of them are even in the prokaryotes. So that answers that question. Okay, going back to our list. So chloroplasts are going to be the site of photosynthesis, okay? And so with photosynthesis, this is going to use the solar light in order to store that in the form of glucose. So chloroplasts are only going to be found in plant cells versus mitochondria will be found in all cells, okay? So that is important to know, an important component of them. Also, the mitochondria and the chloroplasts are theorized to be endosymbionts. So long time ago, it was thought that the ancestral eukaryotic cell engulfed a chemosynthetic prokaryote, basically a prokaryote that could go through this active respiration. And that became the mitochondria. And then the chloroplast was also a photosynthetic prokaryote that was engulfed. And so important to know is that the mitochondria and the chloroplast, they have their own DNA, and it's circular, like a prokaryote. They have their own ribosomes, which are also like a prokaryote. And they don't really need anything from the cell. Like, they are just basically chilling in there. The cell brings in the food and the mitochondria breaks it down, but they're not really needing to get any proteins from our cells. They have their own DNA. They can make all the proteins that they need. Okay, so let's keep going with our list. So vacuole is for storage. There's 3 different types of vacuoles you can have. There's a food vacuole made when you go through phagocytosis. There is a central vacuole found in our plant cells that's going to store water and help with detoxification. We're also going to have a contractile vacuole, which we'll see when you're talking about osmolarity and you're pumping the water back across the membrane. So cilia and flagella, their job is for motility. So cilia is going to be a whole bunch of like smaller segments that are kind of all around the outside. And then the flagella is like a big long tail. So there's a lot more cilia than there are flagella. There's like 1 or 2 flagella. And if you think about a sperm cell, that's a flagella. There's this little flagella at the end of that cell. So that kind of helps you figure out the tail. Same thing with like I wouldn't go tadpole, but you get the idea. So somebody says, is a lysosome considered a vacuole? No, a lysosome is an organelle. The lysosome is synthesized because the rough ER is going to make hydrolytic enzymes, so enzymes that go through hydrolysis, and it then puts it into a transport vesicle that then goes to your Golgi. Your Golgi packages, modifies, and does all that fun stuff. And then when it sends it back out as a transport vesicle, now it's considered a lysosome because it will have those enzymes and is ready to fuse to be able to work with the lysosome to break down the food. Lysosome works with the food vacuole to break down the food. Do we need to memorize the different types of vacuoles? You should be aware of the different types of vacuoles as we move forward into the year because of the fact that those are considered different types of organelles, and you could see them on an FRQ question. Okay, so moving on. Centrioles, this is going to be the part where we have our centrioles. Our centrioles are part of our chromosome movement, so we're going to see a lot more of this when we get into mitosis and meiosis. I wouldn't really worry too much about it right now, but it's kind of the microtubule organizing center. Golgi bodies are going to allow for us to modify our proteins and then package them up. As we talked about before, you're going to synthesize in your rough ER. That's then going to go to your Golgi body before it then moves to the membrane. And last but not least, our lysosome is where we see our enzymatic hydrolysis of our different things. And so this is where digestion is taking place. So when you go through phagocytosis and you bring in food materials, that's how you break it down. If a organelle is non-functional or there's a protein that was made that's non-functional, it'll get broken down in lysosome. If you're in middle school, they always talk about this like as your recycling center. It breaks down all these materials back into their monomers so they can be used again. So like you can break down the nucleic acids that are brought in, and then now you have the monomers of nucleotides to build new nucleic nucleic acids. You can get the monomers of amino acids to make new proteins. You can break it down to get all your stuff to recycle it, if you want to get back to middle school terminology. So saying all that, the questions you're going to have on the exam are going to be more of application based. So example would be Tay-Sachs. Tay-Sachs is a disorder in which the individuals do not make functional enzymes to break down lipids. And due to the buildup of the lipids, they have a lot of the lipids build up in their brain, and they have a lot of medical issues due to that. And so they've always asked, well, what organelle is the one that had the problem? Where are the malfunctioning enzymes at? So the answer would be the lysosome, because we know the lysosomes are responsible for breaking down these lipids. So it's going to be more application. And one of our FRQ questions that we're going to look at actually looks at organelles. So I'm going to let that application just rest for a moment. I do see a question down the little question box, so I'm going to pop that up and see what that says. What exactly is secretion? So secretion just means something being released from the cell. So example would be in your pancreas, you're making the protein insulin. So insulin gets secreted. It gets released out of your cells, and it goes into your bloodstream and travels all around your body, allowing you to absorb the glucose. So I hope that's answered your question. You secrete, basically, if it's leaving the cell, it's secretion. You secrete sweat. You're secreting saliva. Anything that's coming out of those cells are considered secretion. So I hope that helps your question that you had, sweetheart. Okay, what type of cells would have a lot of lysosomes? A cell that needs to break things down. So I would assume your white blood cells, probably your macrophages and your white blood cells probably have a lot of lysosomes, because their job is to engulf bacteria and break it down. So I would assume that the macrophages probably have a lot. Okay, so next part. Our next thing is on membrane transport. Okay. Ooh, I didn't realize when I put this little animation from the Amoeba Sisters that it was actually going to work out. Do all secretion go in the bloodstream after exiting cell? No, there's different types of secretions. So there's an endocrine, which is going to go into your bloodstream, and exocrine, which is going to be secreted out of your body. So for example, where you secrete your saliva, that's considered exocrine because it's going into a cavity, versus the endocrine is going into your bloodstream. And I haven't taught anatomy in quite a little bit, so just nice to me if those terms were not exactly correct, but I believe that that's the difference there, but they're not always in the bloodstream. Anyway, so there's different types of transport we can have. So we can have passive transport, or we can have active transport. So with our passive transport, this is dealing with it. There's no energy required. Okay, so this means that we're going to go down our concentration gradient. So students sometimes have trouble figuring out, well, what does that mean of concentration gradient? So think of it like a hill, right? When you're at the top of the hill, you have a high concentration. Or at the bottom of the hill, you have a low concentration. So if you're going down your concentration gradient, you're starting at this high top of the hill and you're going down. Okay. Versus when I talk about going up your gradient or against the gradient, that's going to be coming from this low region and going against gravity and coming to the top part of the hill. So you'll be going low to high when we talk about going against. But right now we're in passive, which means we're going high to low. And everything wants to move high to low. It releases energy to move high to low. And so that's going to be considered passive transport. There's two types. The first type is simple diffusion. Simple diffusion means that it's going to go directly across the membrane, requiring no type of protein or anything to help it. It just goes in between those phospholipids. Why? Because it's small and nonpolar. Example would be oxygen, carbon dioxide. These are materials that are itty bitty and can fit in between those phospholipids. And then they're nonpolar, meaning they're able to dissolve in that hydrophobic core of your membrane. If you remember, your membrane is made up of phospholipids. And so you have these fatty acid tails in here, and then you have the phosphate heads on the outside. And so here you can see we're moving down our concentration gradient from where there's a high concentration to where there's a low concentration. And so this is considered simple diffusion. versus facilitated diffusion means that I have a protein here. Okay, so there's two different proteins you can have. So this one is considered a channel protein because we just have like this hydrophilic region in the middle and the material is passing through. It's like a tunnel or a channel that passes through so that this ion or this protein or this polar substance is able to pass through. Example would be an aquaporin. Aquaporin is going to be how water gets into your cell. Water will come through by simple diffusion, but it's very, 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 very slow. So most of the water is going to come in through this facilitated diffusion coming through a protein. Another type of facilitated is called a carrier protein. So I always use like a little ball. I don't think I have one that I can use around me, but what happens is that it's going to bind. And when it binds, it causes a conformational change. Anytime you bind something to a protein, it changes shape. So it has a conformational shape change, and as the shape changes, it then brings the material across that membrane. So it came from this side to this side because my carrier protein changed shape. So we always say like you bind and then you carry it across the membrane is a good way to think about it. When I say channels, or am I talking about ion channels? An example of a channel protein is a ion channel. So if I say like the sodium channel or the gated ion channel, those are channel proteins. Yes. Okay, so we said that. Okay, so the other type, other than passive transport, is active transport. So with active transport, it's going to require energy. So we're going to go against our concentration gradient, going from low concentration to high concentration. So you can see we're going from this low concentration to the high concentration. Now, in order to do that, we have to have ATP applied. You have to put ATP in. It has to phosphorylate it. And so when you phosphorylate something, you prime it to do work. Okay, so what's going to happen is ATP is going to be hydrolyzed, so it loses that phosphate, and it's going to bind to that protein. When the protein gets the phosphate, it has a conformational shape change, and it changes shape, which allows that ion to be pushed against this concentration gradient. Same idea as if we were talking about a carrier protein. Usually in your active transport, it is usually a carrier protein because it's going to bind, change shape, and pump the material onto the other side. We talk about it as proton pumps, so moving H pluses across the ion. And we talk about with sodium-potassium pumps. So sodium will get pumped one way and potassium will get pumped the other way, which is how we're going to work out in your nervous system. So, hi, Melanie. If you don't know the Absolute Recap just came in, and she has a great podcast for biology if you need to podcast, y'all. Okay, so we talked about active transport. Okay, so next thing we have is looking at bulk transport. Okay, so this is like kind of big things that aren't going to be able to pass through an ion, a channel protein or carrier protein, things that are not able to come through because it's just too big. Okay, so exocytosis is going to be things leaving the cell. So my students have looked and said, oh, well, exo makes me think of exiting. So like this is dealing with things leaving the cell. Okay, and so with this has to do with like, if we're talking about secretion, right? So when we made the protein in our rough ER that then went to our Golgi, that then went through the transport vesicle to the plasma membrane, it's going to bind to the plasma membrane, the little vesicle, and it's going to open up to secrete the material out. That's exocytosis. We're literally just secreting in this process. Versus endocytosis is when we're going to enter the cell. Things are going to come in. So there's 3 different examples in which you can do with that. Okay. And so the first type we have is called phagocytosis. So with phagocytosis, it's going to be cellular eating. So you have these little pseudopods that are going to extend out and surround that food substance. Okay. When that comes in, it makes this little food vacuole that then fuses with your lysosome in order to digest it. Okay, that's phagocytosis. Penocytosis is just going to be cellular drinking. So it kind of just brings in some of that extracellular fluid, which allows you then to get more liquid inside the cell to digest fun stuff. And then receptor-mediated endocytosis, you have these little receptors. So there's going to be a ligand that binds the receptor, which then causes this little invagination. It kind of makes like a little pit to then create like endocytosis to take place. So those are the 3 different types: phagocytosis, penocytosis, and receptor-mediated endocytosis. And there's the content if that's what you're wanting from me. So there's been a couple questions coming up. Remember, I said that I'm focusing on unit 1. So functional groups was I'm focusing on unit 2. Functional groups was unit 1. So we're not worried about unit 1 right now. And then my slideshow from unit 1 is posted on my Weebly. So if you're needing unit 1 stuff, it's all on the Weebly, I promise. So as last thing is in my students unit 2, but it's actually, I think, in the actual curriculum, maybe in unit like 4 or 5, it's cell communication. And cell communication is going to show up all year long. Now, the problem is that on the exam, they're going to have a specific example. In order for me to give you that specific example, it's going to take a very long time, because every single cell has different proteins within their pathway. Every single cell has different receptors. There are so many different ligands out there. Cells have different responses to the same ligands. And so there's no way to actually prepare for every single pathway you're going to see. So you have to prepare for the generic and be able to apply. So there's 3 steps to cell communication, 3 steps to cell signaling or the signaling transduction pathway. The first step is reception. You're going to receive the signal. So the signaling molecule is called a ligand. Okay, ligand is going to bind to the receptor. So where that receptor is tells you a lot about that signaling molecule. If your receptor is sitting on the membrane, that means your signaling molecule must be polar, because your molecule, your signaling molecule, cannot get through the membrane, so it has to have a receptor on the membrane to bind to it. If your receptor is inside of your cell, that means your signaling molecule must be nonpolar. So the big example we see here is a steroid hormone versus a protein hormone. A protein hormone is going to have a membrane-bound receptor versus a steroid hormone is going to have an intracellular receptor. That means the receptor is within the cytosol or within the nucleus. So the first step was when ligand or signaling molecule binds to the receptor. Whenever you bind to a protein is always going to change shape. Okay, so that is the first step of transduction. The protein change shape. That shape change then transduces the signal. It allows the signal, not the molecule, but the message. The message is being moved throughout the cell. And so my students just did an FRQ recently. It was the 2013 number 8. And it was about cell signaling. And a lot of the students got confused because they thought the molecule was moving through the cell. And that's not what's happening. The molecule stops at the receptor. Then the receptor changes shape, which then allows for the relay molecules to change shape. So why do we need to do transduction? So transduction is going to, number 1, transfer the signal from reception to response. Number 2, it can allow for amplification. Okay, if I tell you one thing, right, and then you tell all your friends, you're going to amplify that message. You're going to make it so it's a lot larger. It's better than just one person telling another person, just having this relay. It ends up just kind of branching out. And so I think there's an example in our textbook that like you start out with like 1 like glycogen molecule and end up with like 10 to the 6 glucose molecules. Like it's huge. It all has to do with that amplification due to transduction. There's 2 main ways that we do transduction. The first one is a phosphorylation cascade. This just means that you phosphorylate the next thing. So there's a protein, important to know the protein name. It's called kinase. Kinase is going to phosphorylate something. So if I have a protein kinase, it means that I'm phosphorylating a protein. And we'll see other kinases later on in the year. And then the other thing we can have is a secondary messenger. So your original signal molecule, your ligand at the beginning, was your first messenger. And so a secondary messenger would be like calcium that's stored in your smoothie R, right? We said that it would be stored in smoothie R. Smoothie R is then going to release that calcium. So the signal comes in, the message says, okay, release calcium. Calcium then gets released, which then continues that transduction pathway. So it's just a small molecule that is in the cytosol that allows for the next process. And another one you could have is cyclic AMP. Cyclic AMP is going to be ATP is adenosine triphosphate, so it has 3 phosphates. AMP is adenosine monophosphate, so it has 1 phosphate. And then that cyclic in the front of it just means that it kind of makes a ring structure. And so cyclic AMP is another one of those secondary messengers you can have. And again, you're just going to see a diagram that will have this stuff on it. You're going to have to connect to whatever you see. I have no way to know exactly what you'll see come the AP exam, or I'm not going to tell my students what's on their exam tomorrow. Somebody says, what does phosphorylate mean? Phosphorylate means just add a phosphate. So when we were talking about our active transport, I said that we were going to phosphorylate that transport protein. That just means we're adding a phosphate to it. You add the phosphate, it primes it to do work. It has a conformational shape change. It has a higher amount of free energy. If you want to think about like Gibbs free energy, by having the phosphate on it, it has higher free energy, which allows it to now do work because it's now an exergonic reaction. If you're already talked about the reaction process, you know those terms. If you don't, that's all in Unit 3, so don't really worry too much if you don't know what exergonic means. I just try to connect some things when I can. Okay, so the last thing you can have after we get through this transduction is our response. So tons of things happen. There's a lot of different options. The cell can grow. The cell can go through apoptosis. The cell can turn on or turn off a gene. There's all kinds of different responses that could happen. It can synthesize something. So you're going to have to look at whatever's in the diagram to make that decision. You'll have to do a reading of the prompt. Sorry, they do give you information in the prompt. You're going to have to read. I know reading is fundamental. So you have to just apply to whatever they gave you. I cannot remember off the top of my head what year it is. I want to say it's like 2017, 2018. There's an immune system kind of question that goes over the making of gastrodermin, or a membrane protein, and then it also talks about interleukin, and it like releases the interleukin out. So there are some practice questions out there that you could use that are like commonly available for you if you're needing to practice signal transduction, and this how you could see on the exam. So here's a picture of what we just talked about. So we have a ligand binds to the receptor, conformational shape change. This right here is reception. So then we would go through this cascade right here. This is my transduction, and then our response is whatever happens because of that. Ryan says it is 2018. Thank you, Ryan. And it would be, I think, question number 1 on the 2018 exam. It's the first question we see. Oh, no. Sorry. Stuart says it's question number 2. Thank you, Stuart. So 2018, number 2. So I have 2 free response questions to go over with you. Just questions that I was looking at today as students were asking me, hey, what would be good questions to study? And so I was flipping through my binders because I just have a whole bunch of binders. And so I was like, okay, I think these would be good for us to go over because they're 2 short FRQs that we can kind of quickly do that won't take too much time process. So the first one we have is the 2019 exam, number 8. It gives you a beautiful diagram, shows you what the pH is before. So pH of 6, and the cell volume is small, and then a pH of 7.7. Now it's large. Overall, what's happening is there's this flower. It changes from red to blue, and the petals are going to swell while the flower opens. The pigment is due to something that's found in the vacuole. So you can see here that there's a vacuole in our pictures. And then the model of how it works before and after the flower opens is in the picture. Okay. So it says, identify the cellular component in the model that is responsible for the increase in pH of the vacuole during flower opening. So if you look here, you can see, okay, well, before it opened, we had a 6.6, and afterward we had a 7.7. So what in your picture, remember, a lot of times you can get the answer from the picture. So what in the picture is allowing my pH to increase? So you first need to think about something. If my pH is increasing, which this is a Unit 1 topic, I know. If your pH is increasing, that means that you are going to decrease your hydrogen ion concentration, because pH is looking at the concentration of hydrogen ions. And so it is a negative logarithm. So when your pH increases, it means that your H pluses are going to decrease. So if you look at your picture, where do we see those H pluses leaving from? So you see that we have a whole bunch here, we have a bunch here. What's happening to allow for this to go from there to there? Okay, well, what's happening is that we have this transport protein right here, right? So this transport protein is allowing these H pluses to leave. And as they leave, it changes the pH, okay? It's going to make it more basic, okay? And the description would be because it lets the H pluses out of the vacuole. That right there is 2 points. Just being able to look at this diagram and go, hey, look, we had 3 H's. Now I have 1 H. Oh, what happened? Oh, it must have went through this thing right here, okay? Let your diagram tell you what to do, okay? Apply the biology that you know to the diagrams because you know this. You all have this. This is right in front of you. You can do this, okay? Remember, you're penguins. You're dressed for success. So the second part, did I answer the rest of that question? I did, forget. So the second part of this question says that we have a researcher claims that the activation of the transport protein causes the vacuole to swell with water, provide reasoning. So first thing you can look at is you can say, okay, well, here's small and here's large. So when it opened, now I see that this has a higher amount of volume. So what had to happen? Why did this swell with water? So if you look, you can see that we have a whole bunch of ions in here that we weren't in there before, okay? So now your vacuole has a high amount of ions. It has a high solute concentration, which means that it's going to be considered hypertonic. And since it's hypertonic, the water is going to flow into that area. And so your answer would be that the concentration of the solute is increasing inside the vacuole, or the solute moving inside the vacuole makes it hypertonic. You could also say that it lowers the water potential. You could also use the words hyperosmotic in case you're needing that. So I hope that was helpful. And the second FRQ I have to go over with you is about organelles. So remember I said that they're going to be more of application questions. It's not going to be a straight, what is the function of this organelle? It's going to be application based, okay? And so what you've been given, go backward, machine. Really? You're really not going to go backwards for me? Okay. So they've given you 3 different cells, and we're going to break it down just to make life easier. But here's the full-on question. It's 2013, number 6. And so they give you smooth ER, rough ER, mitochondria, cilia, and Golgi for each of those. And your responsibility is to look at what the cell has and determine what the function of the cell, what the function of the cell is, and explain how that data supports that identification. Okay, so let's look just at X. So if you look at X, there's a small amount of smooth ER, okay. There's a small amount of rough ER, okay. But there's a high amount of mitochondria. So if you remember, the job of the mitochondria is to synthesize ATP. So this cell must need a high amount of ATP. It must need a bunch of energy in order to do something, right? And then if we continue looking, we see that we have cilia present. So that means that there's going to be some type of movement, right? And then the Golgi bodies, there's a small amount of those. So the big thing that we're looking at here is that it has mitochondria and cilia. Well, what does that mean? That must be in charge of movement. The cell must be part of locomotion or movement. And it's because of the fact that it has the cilia for the movement and it has a large amount of the mitochondria, which is needed to provide the energy for that process, okay? You have to get both points to get the 1 point. You have to say everything that's in that row in order to get that point. You have to give me both the function and you have to explain it, okay? Be very careful that you're not just name dropping things and going, oh, well, it has this. Be very, very clear and demonstrate that you understand what you're talking about, okay? So Y is another difference. So here we see that we have a large amount of smooth ER. We have a large amount of rough ER. There's not so much mitochondria, not too much cilia, and then we have a large amount of Golgi. So this can go 2 different paths, right? So we can either go the path of, okay, well, the smooth ER's responsibility is detoxification, right? So you could say detoxification is the function of the cell. You could say this for lipid synthesis or hormone synthesis. And the reason why is because the smooth ER, there's a large amount of smooth ER, which is responsible for making lipids and hormones, okay? You could go the other path, looking at the rough ER and the Golgi, and say, okay, well, rough ER is going to make those proteins for secretion, and Golgi bodies are going to package and kind of modify them. And so it could be for secretion exotosis, it could be for protein synthesis, and it's because of the fact that it has that large amount of rough ER and Golgi, which is required to produce the proteins and, of course, package those proteins, okay? So again, it is a very descriptive answer, a very thorough answer on this, okay? Okay. Somebody says, are the terms ATP and energy interchangeable for these kind of questions? Yeah, I say they are, because ATP is kind of the common term for energy. So as long as you can demonstrate that you understand that it is synthesizing ATP, you can't really say that it's synthesizing energy. So I guess maybe it's not interchangeable, because energy can't be created or destroyed. The energy is coming from something, and that mitochondria is using that released energy to fuel the adding of a phosphate to ADP, which we'll learn more about in Unit 3 when we talk about cell respiration. So the last one is the one that students never get. And so this one is weird. It is missing all of the organelles. You don't have smooth ER, you don't have rough ER, you don't have mitochondria, you don't have cilia, you don't have Golgi. It doesn't have any organelles. What am I supposed to do with that? Well, a lot of our cells, they are needed to not have organelles. So for example, your red blood cells, their job is to bring oxygen around your body. If your cell, your red blood cell, had a mitochondria, it's going to use that oxygen up and won't be able to transport around your body. Plus, by having no organelles, it increases the surface area, so now your red blood cells can carry more oxygen. Another example, if it's a dead cell, if the cell is dead, it doesn't need any organelles because, well, it's dead. So like the tracheids that are in your plant cells that are allowing for the water to move up the xylem. So that's an example of another type. So there's a lot of different things you could put on the answer choice for this one. You can talk about that it's for transport. So talking about oxygen transport or water transport in plants. And the reason why, because it doesn't need those organelles. Talk about protection. So like your epidermal cells, again, don't require it. Support, talking about the gland tissue or the vascular tissue. Again, you don't know these words, so whatever. Doesn't require. You talk about storage, talking about how it maximizes the volume or the space available, like your hemoglobin. Again, doesn't need the organelles. And then it could have no function because, well, it's a dead cell. Because it doesn't require those organelles. So, are there any other questions before I log off this system? Because that is my last one that I have. While I wait for your questions, I'm going to remind students that I do have those quizzes games. These are the codes that are currently active for anyone in the world to do. By the time that you're studying for your AP exam, there's a chance that these codes might not be active anymore because of the fact that I have a maximum of 500 people that can take it, and I've already maxed out, I know, on water, on my water one. So I had to redo that one, but so we should be able to. What are the concepts that we need to know for the test tomorrow? So the test is going to be broken down between, then I'm talking about my unit exam, not the AP exam. So our test is going to have chapter 4, 5, and then like 5.6. And it's going to be equally distributed between all of those. You'll have 30 multiple choice questions and 2 numeric responses. You'll have 5 short FRQs because 2 of them are being treated like they'll make a long one. Did I say something wrong? Yes, Unit 2. We are in Unit 2. We're not in another unit if I said the wrong chapter. I'm sorry, y'all. And so I think it worked out to be about like 9 questions from chapter 4, like 11 questions from chapter like 5.1 through 5.5, and then like whatever's left is 5.6. Because cell communication is a big part of the AP exam. So I think that I've answered that. One of y'all sending me pictures, and I don't know what you're doing. So Stuart says Unit 2. I don't know if I messed up something. Good content, thanks. I think my class is behind them. That's okay. I'm really far ahead. Our school does start in August. So I've been with my students for well over a month. So we've had plenty of time to get through our content. Plus my students are going to be done probably in March. Describe the function of a secondary messenger. Yeah. So a secondary messenger is just going to be a molecule within the cell that can diffuse to then continue the transduction pathway. So it would be like, okay, think about a Rube Goldberg experiment, right? So you have like a whole bunch of dominoes that go down, right? And then at the end of the dominoes, there's like this ball that like bounces, and the ball goes and sets another set of dominoes off. That bouncing ball is like your secondary messenger. It's moving over to another part of the transduction pathway to continue it. I hope that helps you. There's 2 different types. You could have calcium, or you can have cyclic AMP. And again, you'll just see it in the picture. So they'll have to give you some type of background information. They can't just expect you to know what pathway they're talking about. Every single cell communication question I've ever seen always tells you some type of information, and you have to apply it. So are there any other questions that we have? Going once? Going twice? Sold to me going to get on the treadmill. Okay, so remember, you are AP Bio Penguins and you are dressed for success. Good luck to my students on their test tomorrow, and good luck to anyone that is watching this later as you prepare for the AP exam. Bye, y'all.